Yes, children. So in the yesterday's class, we are done. Record. So in yesterday's class, we are done. The DNA is the genetic material, and we have removed all other contestants because they were not fitting into our criteria of genetic material. And then we also studied central dogma. What did central dogma said? That a DNA can replicate itself, a DNA can transcript itself into RNA, and RNA can translate itself into protein. And it is these proteins, some of these proteins may act as enzymes, and these enzymes will be able to control various kinds of activities in the body. So this is the way of working of any function in our body. Okay, DNA to DNA, that is replication, DNA to RNA, that is transcription, RNA to protein, that is enzyme. That is called central dogma. Now, when we were talking about DNA as a genetic material, so to make DNA to RNA we require some enzymes, and to make RNA to protein we require some enzyme. Now the question is, DNA directly cannot make it protein, and protein cannot be made as long as DNA is not there. So it is a situation that who came first, whether it is DNA came first or whether it is protein came first. And if it all protein came first, how they have come first? Because proteins are actually made by means of the central dogma, and if it is DNA came first, then how DNA replicated to make protein? So it is a very complicated question on which scientists were trapped for sufficiently long time. They never had an answer to it. Then they came to know there is another species of nucleic acid called RNA. There is another species of nucleic acid called RNA. And RNA has an ability of self-replication as well as self-catalysis. Self-catalysis means that is RNA does not require any enzyme as protein to control their activities or to make proteins or show their characters. So it was believed that the RNA must have come first. RNA can make protein without using protein enzyme. These reactions can be catalyzed by certain kind of RNA itself. So that means RNA don't require protein, and that is why the RNA can make protein, and protein can carry out the various type of activities which they require in the body, and that's why RNA can easily express themselves. They do not require any other thing in between. So this was a very good experiment or property which we have found in the RNA. So it is believed that the RNA was the first genetic material. Even today, there are several viruses in which RNA is found as a genetic material, and they are very good controlling their activity. RNA to protein, RNA to even make DNA, and DNA again make RNA, and RNA again make protein, and all as and when they require, those processes are controlled. So that means RNA can serve this purpose better than DNA. Now the question is, if RNA can serve the purpose better than DNA, then what is the need of DNA? So yesterday only we discussed that both RNA and DNA are self-replicating, but RNA is failed on the basis of or on the criteria of stability. RNA is not chemically stable. RNA is not physically stable. When the temperature increases, RNA spoils. When certain chemicals are exposed to RNA, RNA degrades. That is why RNA cannot be made as a genetic material in most of the organisms. It is only genetic material in certain organisms, the lower organisms, which may not be exposed to this type of condition. Or even if they are exposed, they are so many that it hardly affects. They will not die, but they will not discontinue. Okay, so their very survival is not affected. So all the initial organisms were having slowly the number of protein increased, and then came DNA. And then DNA started converting, making RNA, and RNA started making, and that is what the today's central dogma is in being. So today we find that the genetic material is DNA, DNA make RNA, and RNA make protein. But earlier time, how first the protein must have come. So for that the information must have come like this. So the not the protein but RNA came first, and DNA came quite later. Okay, so this is what the hen and the egg problem solution. Talking about RNA, what are RNA? You all know already that RNA are also a species of the nucleic acid called ribonucleic acid. The difference lies in that RNA has ribose sugar in place of D-ribose sugar and uracil in place of thymine in molecular form. That is when we talk about the molecule. And when we talk about the structure, the DNA are double helical, whereas the RNA is single stranded, and DNA is always helical, whereas RNA have three different forms: mRNA, rRNA, and tRNA. mRNA is straight fibrous structure, whereas the tRNA is clover leaf thick. RNA is three species: mRNA, tRNA, and rRNA. Whereas the mRNA is thread-like structure, the tRNA is clover leaf shape, and rRNA is globular, and the rRNA will make ribosomes. Okay, they are globular, so structure is different. The type of RNA molecules are there and the proteins are there, they differ. So RNA is quite different from DNA. The differences we already done in class 11. Okay, so that is the thing. So DNA is only and only one type. RNA is of three types, and all three type of RNA has different function. Whereas the messenger RNA is acting as a blueprint of what kind of protein has to be made. tRNA is carrier which brings the required type of amino acid and give it to the ribosomal RNA, so that ribosomal RNA can catalyze them and required type of proteins are formed. And ribosomal RNA or rRNA are actually the molecule which catalyze them, which join them, and which make the long chain of polypeptide, that is protein. So there are different functions, different structure, different type, but RNA is much smaller than the DNA. If we consider the molecular weight, the DNA's molecular weight may go in several thousand, whereas the RNA's molecular weight can as small as some hundreds to some thousand. So all the type of differences are there. Structurally they are different, whereas the DNA is double helical, RNA are three different type of structures are there. Molecularly, like if we talk about the chemically, then the RNA is ribose sugar, DNA is deoxyribose sugar, RNA is uracil nitrogen base, whereas the deoxyribose has thymine as a nitrogen base. RNA do not form any bonding between the two nitrogen base, that is the purine and pyrimidine, whereas the DNA is making that. RNA is less stable in compared to DNA. DNA is more stable because their both strands are covered with each other, that is the complementary to each other. The size RNA are much smaller in compared to DNA, and function we already done. Further detail about RNA we will study later when we will discuss about their role in protein synthesis. Okay, so this is what the thing is. Now we shall study replication. Replication. What is the replication mean? Replication means making a new DNA from an existing DNA. So this is exact copy. very precise, very genuinely copied. There is no chance of error. But once in a million there may be an error possible. We cannot say that it is total error-free mechanism. Definitely errors are there, but the errors are not like human error, where we keep like we do something and we commit lot of errors. But these are total chemical processes and the chances of error are much less. Now how it is done? Replication of DNA was, the idea of replication of DNA came in the mind of Watson and Crick when they were giving the structure of DNA. When they were giving the structure of DNA, that they found that these two strands are same. Only thing is that they are antiparallel, but they are chemically stable and same. And what they found that adenine always combined with thymine, thymine always combined with adenine, cytosine always combined with guanine, and guanine always combined with cytosine, adenine to thymine, thymine to adenine. That means if one strand is known to as second strand, we can easily guess. If one strand is open like this, if the two strands of the DNA are open like this, here it is A, here it is A, here it is C, it is G, it is T. It is obvious that the new nitrogen base or nucleotide which will come and attach here will be only T. Only T type of nucleotide, only G type of nucleotide, only C type of nucleotide, only A type of nucleotide. And here it was T, here it was T, here it was G, here it was C, and here it was A. So T will always have A type of nucleotide, T will always have A type of nucleotide, here it will be C, and here will be G type of nucleotide. Because the base pairing rule is so confirmed, so error-free, so fixed that no other nitrogen base can go and fit into these places. So Watson and Crick suggested that means the DNA can self-replicate itself genuinely, faithfully, 100% error-free. So that was one of the very important property which we have found. When studies were going on, we came across that the process to replicate DNA will require a number of enzymes. First we studied what the enzymes. There are 5 different kinds of enzymes required. These are helicase, topo, isomerase, DNA dependent, DNA polymerase, DNA dependent ligase, and primase. We shall study the function of each one of these. This is our DNA. Just like a rope, the electric wire, you might have seen the electric wire which comes, coiled one. If you try to open them, stretching, you will find that it is not easy. Because as you start pulling here, the downward it will become all the more, the coiling will be all the more strong. Helicase enzyme is responsible for uncoiling, separation of the two strands. Separation of two strands. But the two strands you cannot separate, because as you are pulling it apart and trying to separate it, it will become all the more coiling at both the ends. So what has to be done? Then we require topoisomerase. What will topoisomerase do? It will cut it from one of the points. As it is cut, the coiling can be uncoiled, and then again it can be joined. So it removes the coil, the force of the coil, which is encircling or whatever. That is removed by. Third enzyme is primase. We will study about primase a bit later. Fourth one is DNA dependent DNA polymerase. DNA dependent DNA polymerase is an enzyme which always make the copying in DNA. This is the DNA which we open. This is called replication fork. Here it is. I'll explain how it goes. Just now you just understand the importance of DNA dependent DNA polymerase. In yesterday's class, or rather day before yesterday's class, we have studied the structure of DNA, and we studied that each strand of the DNA is running anti-parallel to each other. If one is having 3 to 5 moiety, if it is having 3 ends, so this will be 5 ends. If it is 3 ends, so it will be 5. The new strand always move in 5 to 3 direction. So new strand can only be formed on the existing 3 to 5 direction strand. Suppose it is T here, T here, A here, C here, G here, A here, C here, G here. So it will be C, it will be G, it will be T, it will be C, it will be G, it will be T, it will be A, and it will be A. So when the replication fork is formed, the nucleotides are already there in the cytoplasm. Automatically on the 3 to 5 strand, the new nucleotides will come and arrange on it according to base pairing rule on the 3 to 5 end. So A will have, T will have A type of nucleotide, T will have A type of nucleotide, A will have T type of nucleotide, C will have G type of nucleotide, G will have C type of nucleotide, and they will be continuous and they will form a continuous chain. But on the other hand, the chain which is running from 5 to 3 directions, since the new chain can only form in this direction, and here the uncoiling is still not taken place. So a very simple mechanism is developed. This is controlled by an enzyme called DNA dependent DNA polymerase. And for this, what happened? There are some primers already made and kept, TTA, some 3, 4, 5 type of single stranded DNA molecules. They are already made and kept. So on the TCG, it will be AGC, like that. And they will, on the basis of complementary rule, they will come in a range of that. And the bond will form between that. Double one, double one, double one. So primase, what they do? They make small stitches of single stranded DNA known as primers. Primase enzyme makes small stitches of single stranded DNA with random nucleotides. And in a cell which is undergoing DNA replication will have thousands and thousands type of such combinations of the primers available. Now, whenever a nucleotide open, if complementary nucleotide sequence is there, automatically the primers will go and arrange on them. And then the function of DNA ligase comes, which seals them together, which will join these strands. So 5 enzymes are there. First is helicase, second is topoisomerase, third is primase, fourth is DNA dependent DNA polymerase, and fifth is DNA ligase. Let us study how the process of replication of DNA takes. It is a very simple process, not a complicating or difficult. This you can understand in once if I explain it to you. चलिए यहाँ पे पढ़ते हैं. We got a DNA molecule. Helicase and uncoil them and topoisomerase release the force, coiling force, and a replication fork is formed. This is called replication fork. Now, suppose this is the strand in which 3 ends of the sugar phosphate is deoxyribose. So this will be 5 end, and this is the 3 end. So it will be 5 end. Here 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4. Okay, here we'll write some nitrogen bases. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. And here if it is A, here it should be T. T, A. T, A. G, C. C, G. C, G. G, C. E, A. A, T. A, T, T, A. Which two enzymes did this function? First enzyme was helicase, which uncoiled them by separating the hydrogen bond, and second topoisomerase released the tension. Topoisomerase, what it did? It released the tension. Okay. DNA replication will always begin from a point. That point is known as point of origin. It can be in between anywhere, or it can be towards one end, or it can be towards another end. It is always fixed. For a DNA, it may have one or it may have more than one point of origin, but whatever, it is always fixed. But just for the study of ease of understanding, we have taken the point of origin towards one end, so that the replication path is separated. Had it in between, then they would have simply uncoiled it. Now what happened? Now what happened? This is 3 to 5. So no sooner A type of nucleotide open, T type of nucleotide came there and formed a bond. A, T nucleotide came and formed bond. A, T nucleotide bond came and formed bond. And these are sealed by DNA-dependent DNA polymerase enzyme. Interestingly, in our body, wherever energy is required, what we use ATP, adenosine triphosphate. Here ATP are not required. What is required then? Triphosphonucleotides. Thiamine triphosphate. Thiamine triphosphate. ठीक है? Thymosin triphosphate. Adenine will have, thiamine will have adenosine triphosphate. Guanine will have cytosine triphosphate, and triphosphate will lose 2-phosphate. 2-phosphate they lose, and the energy of these 2-phosphate will be used for catalyzing the process. And 1-phosphate it will have it. So what I want to say, it is a continuous process. As the nucleotide open, and corresponding nucleotide will come there. As the nucleotide open, corresponding this come, and they will be sealed there and there, catalyzed there and there. Seal means catalyzed. So this way. So this is called continuous strand. What it is known as? Continuous strand. This is known as continuous strand. It will go on one after another, non-stop. On the other hand, the strand which is running from 5 to 3 direction, the new strand also has to run in 5 to 3 direction. So for this, either this chain fully should open, and if it fully open, then the process may be like it may totally gets lost or something. So it is not advisable. So what happens? Some primers are already there. These primers may have different combinations or 2 or 3 nucleotides. A, T, C, G, A, T, A, G, C, G, and all. Hundreds of combinations. Okay, now what we have to do is, no sooner like triple A is there, we have got a strand, this will fit, this will come. Okay, so if we have got such primer which has 3A and 1T in 3 to 5 direction, that will come and fit here. this will form a strand. Then here, if we got such type of primer there, which are G, C, C and G type of primer, this will, and the DNA dependent DNA ligase will join it. So this process will continue. Since it is not a continuous one, it is stopped number of times. It is lag behind. This is a progressive. This is lagging behind. So such type of strands are also known as lagging strands. Lagging strand. The primers, unless they are joined together, they are called primers. Once they go and attach on this strand, then they are known as Okazaki fragments. So these are now known as Okazaki fragments. Okazaki fragments. So this is how the DNA replicates. A DNA enzyme replicate will break the hydrogen bond between adenine and thymine, cytosine and guanine, and topoisomerase will uncoil and replication fork will form. Two strands will be there, they are served as the template strand. One will be going 3 to 5 direction, other will be going 5 to 3 direction. New strand will start forming on the 3 to 5 direction, and the new strand always progress from 5 to 3 direction. So adenine open, immediately thymine type of nucleoside will come. Then adenine open, thymine. And what molecule are these which are coming there? 3-phosphate containing nucleotide. So it is thymine, we will say thymosyne triphosphate, adenine triphosphate, guanosine triphosphate, cytosine triphosphate. And when these triphosphate comes and arrange them, 2 phosphates energy is used for bonding, and 1 phosphate will remain there, that will be extending the length of the new DNA strand. Since it is continuously progressing one after another, it is called continuous strand. On the other hand, the strand which is having 5 to 3 direction, new strand also has to run in the 5 to 3 direction, which is not practical. So they have once a certain stretch of the DNA is open, a strand is open, then it serves as a template for some of the primers. Primers are made by enzyme primase. Primers are single-stranded nucleotides having small number of nucleotides, that is 2, 3, 4, 5, 6 numbers are typed on them. And when a complementary space is open there, immediately they go and stay there. And DNA ligase will join these fragments. These fragments are now known as Okazaki fragments. So this is how the process will begin, and this will lead to the other end. So the two separate DNA will be formed, which are exactly similar. You can see A to T, 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 T to A, T to A. This nucleotide DNA strand will be exactly similar to this DNA, and that is what called as replication of DNA. Very genuine process, very faithful process. There is no chance of error. However, there may be some possibility of error 1 in 1 million. So accurate. If I ask you to write one word even when we punish children, I will not speak, write 100 times. So somewhere the errors are there. When a child is asked to copy a formula number of times, the formula becomes something else. But here, the copying is so accurate that there is no mistake, there is no error anywhere. Okay, so that's all. Thank you for today, and we shall have next class on Monday, as Saturday is the problem solving day.